Hello, my name is Justyna Mika. I'm from Silesian University of Technology, and today I would like to present you the results of the modeling of the proportion of functional T-cell receptor genes with age. T-cell receptors are part of the immune system, which is disseminated throughout the body to provide protection against infectious and non-infectious diseases or trauma. We can distinguish innate and adaptive immunity, both consisting of cells collaborating with each other and ensuring maximum protection. Today, I will focus on adaptive immunity and T cells. T cell activation depends on its antigenic T cell receptors, which are placed on the surface of a cell. Every cell contains thousands of receptors of one type, specific to only one or a few antigens. Thus, maximum protection requires a large number of lymphocytes and a large number of different T-cell receptors. VTJ recombination is a process that ensures this huge variability of T-cell receptors. It consists of the selection and assembly of one of many germline variable, diversity, and joining gene segments into a single exon. This created exon is coding for the variable domain of the receptors and consists of complementarity determining regions, which specifically bind with antigens. The expression of T-cell receptor is acquired in thymus. Two case scenarios may be considered. When a created exon is functional, it is in an open reading frame, then second locus maintains its germline sequence. In other case, the created exon can be non-functional in first locus, and the rearrangement on the second locus might be functional. Thus, after performing targeted TCR sequencing, the ratio of functional to non-functional sequences would be about 2:1. So the goal of this project was to determine if the proportion of functional and non-functional sequences changes with age. This proportion serves as a measure of the dynamics of T lymphocytes population. The data of T-cell receptor repertoires were downloaded from Adaptive Technology website. In that project, blood samples from almost 600 healthy individuals were taken, for which T-cell receptor sequencing was performed, focusing on complementarity determining region 3 of beta chain only. First, we performed the data curation with detection and filtration of outlying samples, including samples with missing information about age or sex. Also, sequences with unidentified V or J genes, orphan genes, or indexing errors were filtered out. As a result, 487 samples were left for further analysis, comprising of altogether almost 2 billion TCR sequences. The age of donors ranged from 1 to 74 years old, and the sex was more or less balanced. To describe the proportions for every donor, further denoted as status diversity, Pielou's J-index with Pacharin's estimator of entropy were used. Pielou's index gets values from 0 to 1, where the higher the index, the more even are proportions of functional and non-functional sequences. Next, piecewise linear regression was applied to find different patterns of evolution in age categories. We used brute force technique by testing all possible age splits, and the best age split was determined based on a minimal Bayesian information criterion value. Also, the model that best describes the data was chosen using PIC value. Coming to results, the best age split was estimated between people of 18 and 19 years old. The obtained models were different for both age categories. For young donors, we observe a significant drop of status diversity. Where the size of an effect is strong, the correlation gets value of 0.6. For older donors, we do not observe a significant change with time. Based on a correlation value equal to 0.06, we can say that the size of an effect is negligible. This drop of status diversity means that there are bigger disproportions between functional and non-functional sequences. So in younger donors, we observe an increase of functional genes, whereas in older donors, it stays stable over time. We can also observe that the slopes of two created models are significantly different. To conclude, we observe the best age split between people of age 18 and 19. Until the age of 19, the proportions of functional sequences increases and stays stable afterwards. This result suggests a different pattern of T lymphocyte proliferation with age. This knowledge might help getting a more precise assessment of the effects of aging on immune system. Thank you very much for your attention.